Welcome everybody. Today I have the pleasure to present my work about antimicrobial proteins, induction, molecular action, and therapeutic potential. So, as you know, the antimicrobial proteins is natural products that is play a central role in host immunity, and this is already directed against a lot of microorganisms, including bacteria, fungi, viruses, and parasites. Based on our publication and recent publication, previous and recent publication, we developed a model for the molecular mechanisms regulating the induction of antimicrobial proteins in response to the infection of bacterial pathogens. You see this model how is liable to shock acid or gram-positive bacteria and LPS, lipopolysaccharides, and gram-negative bacteria stimulate toll-like receptor 2 and 4 to enhance the expression of one of the antimicrobial protein candidates, and that is already characterized in details in our laboratory and published in several publications. This protein called is 100A15 and use it as model for oral infection associated with the induction of antimicrobial proteins. First, we started with the investigation of the expression of this protein S100A15 in response to oral microbial pathogens or the purified components of their membranes. We use it to cell lines derived from oral mucosa and gingiva fibroblast as model for oral tissue. As you see, the stimulation of both cell lines GF, this is gingiva fibroblast, and KB1 mucosal melanoma with lipotoshoric acid enhances the expression of S100A15 at the RNA level. The expression was first noted 12 hours following the stimulation and increased thereafter up to 72 hours in both cell lines. Also, the same results was noted by the stimulation of LPS, the purified membrane components of gram-negative bacterial pathogens. Also, we used the heat-inactivated microbial pathogens from the gingivals. This is oral pathogen bacteria that is common in oral cavity, and sanguines also the other one. The first one, this is gram-positive bacteria, and the second one is gram-negative bacteria. And this is, as you see, this is slot A or in figure A and figure F. And we also confirmed this at the protein level by the stimulation of both cell lines with heat-inactivated microbial pathogens, and you see the expression of S100 in response to the stimulation with the corresponding or microbial pathogens. We addressed also what is a toll-like receptor is involved in the modulation of the infection of gram-negative bacterial pathogens and who is already associated in the modulation of gram-positive bacteria. Using antibody directed against the corresponding toll-like receptor, anti-toll-like receptor 4 antibody and toll-like receptor 2 antibody. And we could show that is toll-like receptor 4 is responsible for the modulation of gram-positive bacteria or lipoteichoic acid-induced expression of antimicrobial protein S100A15, while toll-like receptor 2 was responsible for the modulation of gram-negative bacteria. This is gram-negative bacteria is modulated via toll-like receptor 4 and gram-positive bacterial pathogen is mediated through toll-like receptor 2. Also, we characterize it further. Characterize it further the toll-like receptor 4 and 2 induced signaling pathway, including TREF6 and SK1, Jun kinase, and B38. And as you see, the stimulation of the corresponding bacterial pathogens, or the stimulation of the cell line with the corresponding microbial pathogens enhances the expression of TREF6 in both cell lines, and this expression, the increased expression of TREF6 was associated with the phosphorylation of SK1, Jun kinase, and also B38 as detected by the corresponding phospho-specific antibody. The next, we look at whether this activation of the signaling pathway is associated also with enhancement of their DNA binding activity. Their DNA binding activity of the transcription factor as physiological substrate of the signaling pathway, Jun kinase. And also we look at the physiological substrate of B38, this is ATF1, the transcription factor, and see this is the enhancement of the DNA binding activity of the transcription factor ATF in response to the stimulation of both cell lines with either gram-negative or gram-positive bacteria. Also, the characterization or investigation of the components of the signaling NF-kappa signaling pathway showed also that is phosphokinase or E-kappa inhibitor, the phosphorylation of E-kappa inhibitor in response to the stimulation with the microbial pathogens in both cell lines, and also the degradation of E-kappa alpha inhibitor, the inhibitor of NF-kappa, and also the enhancement of the DNA binding activity of the transcription factor in NF-kappa in response to gram-positive and gram-negative bacteria. Then we use it inhibitory experiments to determine exactly whether these identified pathways is already involved in the transcription regulation of the S100A15. We used SP60125 as inhibitor of John kinase and SP203580 as inhibitor for B38 and B117082 as inhibitor for NF-kappa, and we could show that all the three pathways are involved in the regulation of antimicrobial pathogens, gram-positive and gram-negative bacteria pathogens in the induction of S100A15. We come back to our model, and you see the stimulation of toll-like receptor 4 and 2 led to the activation of three parallel pathways. The first one, all already mediated through TRER6. The first two pathways, including MAP kinase, this is regulated through apoptosis signal regulated kinase 1, leading to the activation of their downstream MAP kinase 3,6 and 4,7, and 3,6 led to the phosphorylation of B38 to enhance the DNA binding activity of the transcription factor ATF2, while the MAP kinase 4,7 enhance the phosphorylation of G and K. This is AB1 site, leading to the phosphorylation of AB1, and the other pathway is already mediated through TREF6, is this NF-kappa pathway, and you see the enhancement of the DNA binding activity of NF-kappa together with the other two transcription factor build out the complex of the transcription complex of S100A15. Next, we provide our data or in vitro data in patient sample using immunohistochemistry and also RT-PCR, and we see this is the enhancement of the S100A15 in patient gingiva with inflammatory gingival, and this gingival associated with the infection with oral bacterial pathogens. Next, the molecular action of antimicrobial proteins in bacterial pathogens is mediated by different models. This different model is already recognized and reported in several studies. The first one is Barl-Steph model. This is, you see, this is binding or the interaction of antimicrobial protein to perform pores in the bilayer membrane, lipid membrane of bacteria. And this other one is Freudel pore model, that is cover or bind, interact with the outer and inner membrane complex and from all sides of the membrane to increase the permeability of bacterial membrane. The other one is the penetration of the antimicrobial protein. This is Carbet model to damage the membrane, and the other one aggregate model and is already the involve the antimicrobial protein and increase spore. Leading all this model is expected to increase the membrane permeability, enhancement of membrane penetration, and interference with cellular components. The mechanism of antimicrobial induced inhibition of cell maintenance and growth machinery is mediated through different mechanisms. First one, this is interference of antimicrobial protein with RNA and DNA to induce DNA and RNA damage. The second one, interference with cellular enzymes to prevent the binding between this is as competitive inhibition to prevent the interaction or the binding of enzymes to their corresponding substrates. The third mechanism in the cytoplasm, this is the inhibition of the transcriptional mechanism or machinery on the cell. All these three processes lead to cell death or bacterial cell death. The therapeutic potential of this antimicrobial proteins, including S100, E15, and you see this is a couple of already known antimicrobial or published antimicrobial proteins, their source and indication of their indication and administration, and the clinical trial, most of them in phase 2 or 3 and already under investigation for clinical application. Conclusion: antimicrobial peptides are characterized by their broad spectrum of antimicrobial activity and are powerful regulatory innate immunity. Antimicrobial proteins have strong cell-killing efficiency, particularly multi-drug resistance bacteria. Antimicrobial peptides offer an alternative approach to overcome the antibiotic resistance of most microbial pathogens. Antimicrobial peptides may be able to overcome the limitation of current antibiotics due to their antimicrobial activity, but the limitation of these antimicrobial proteins, their poor stability, toxicity, and unexpected adverse effects would limit their clinical application. However, the more study and investigation and characterization of their mechanism may help to develop antimicrobial therapeutic strategy to be valuable for clinical applications. Thank you for your attention.